In the vast, glittering constellation of anti-aging science, few stars shine brighter, or more enigmatically, than the sirtuins. These proteins, dubbed longevity genes, are part genetic switchboard, part molecular janitor. They sense cellular stress, modulate metabolism, repair DNA, and orchestrate gene expression in response to environmental cues like calorie restriction. But their performance doesn't happen in isolation. Sirtuins depend on a backstage pass, NAD. Without it, they sit idle. With it, they become maestros of metabolic adaptation. That's where resveratrol, niacin, and fasting step in, each a different instrument in the same biological symphony aiming to extend not just lifespan but health span. Sirtuins, specifically SIRT1 through SIRT7 in humans, are NAD dependent deacetylases and AD-pyribosyl transferases, meaning they regulate other proteins by removing acetyl groups in NAD dependent reactions. This might sound like boring biochemistry, but the downstream effects are anything but. When activated, sirtuins modulate the expression of genes involved in inflammation, mitochondrial biogenesis, glucose metabolism, fat storage, and DNA repair. They promote cellular resilience under stress and help orchestrate the cellular response to scarcity, making them central to the biology of calorie restriction, which remains the most robust intervention known to extend lifespan across multiple species. But rather than live a life of perpetual hunger, researchers have long sought sirtuin-activating compounds, stacks, that could mimic the effects of calorie restriction without the gnawing stomach. Enter resveratrol, a polyphenol found in red wine, grapes, and peanuts, and the original poster child for sirtuin activation. Resveratrol was shown to activate SIRT1 in vitro, sparking enormous excitement and an avalanche of research. While early human trials were mixed, partly due to resveratrol's poor bioavailability, more recent studies using higher doses or better delivery methods have shown improvements in mitochondrial function, insulin sensitivity, and inflammatory markers. Resveratrol doesn't just poke the sirtuins. It also stimulates AMPK, a master regulator of cellular energy, and increases PGC-1A, which promotes mitochondrial biogenesis. In effect, it mimics a fasting signal at the genetic level, telling cells to clean up, conserve energy, and prepare for stress. But resveratrol is only part of the equation. Sirtuins require NAD to function, and as we age, NAD levels decline, a phenomenon linked to mitochondrial dysfunction, neurodegeneration, and loss of metabolic flexibility. That's where niacin and its advanced derivatives, nicotinamide riboside, NR, and nicotinamide mononucleotide, NMN, come in. These compounds are precursors in NAD salvage pathway, helping restore intracellular NAD levels and, by extension, sirtuin activity. Animal studies with NR and NMN have shown improved mitochondrial health, better glucose tolerance, and even extended lifespan in some models. In humans, NR has been shown to safely elevate NAD levels and improve markers of cardiovascular and metabolic health, particularly in older adults. Interestingly, niacin's effects are not limited to being a simple NAD donor. Nicotinamide, a form of niacin, also interacts with sirtuins more directly, but not always helpfully. At high levels, nicotinamide can actually inhibit sirtuin activity by binding to the NAD, consuming site on sirtuins. This makes dosing, timing, and form particularly important. A nuanced approach might involve supporting NAD synthesis through precursors like NR while minimizing sirtuin inhibition by avoiding excess nicotinamide or allowing intermittent depletion through fasting. Speaking of which, fasting is the original stack.
Fasting naturally boosts NAD
levels, activates AMPK, stimulates autophagy, and induces a host of protective genes, many under the control of sirtuins. During energy restriction, SIRT1 and SIRT3 are activated, leading to enhanced mitochondrial efficiency, increased fatty acid oxidation, and suppression of inflammatory gene expression. Fasting also suppresses insulin and mTOR signaling, both of which are associated with aging and chronic disease when chronically elevated. In other words, fasting flips the genetic switch from growth and store to repair and survive, and sirtuins are central to that transformation. What makes this landscape especially compelling is how interconnected these interventions are. Resveratrol amplifies the effects of fasting. Fasting raises NAD, which feeds sirtuins.
NAD
Precursors make sirtuins more responsive to polyphenol. This is not a one-pill solution, it's a network effect. And while most of the research has been done in animals, early human trials and epidemiological data suggest real promise. Longevity isn't just about staving off wrinkles or squeezing a few extra years. It's about maintaining energy, cognitive clarity, metabolic health, and immune balance deep into old age. Still, caution is warranted. Boosting sirtuins without context can be counterproductive. In cancer, for example, sirtuins can both suppress and promote tumor growth depending on the environment. In healthy cells, sirtuin activation is protective. In damaged, pre-malignant cells, ramping up NAD might actually help bad actors survive longer. That's why precision matters, knowing when to fast, how much resveratrol to take, and what form of niacin to use. It's not about pushing sirtuins endlessly, but about modulating them with the rhythm of biology. In the end, sirtuins are less like a fountain of youth and more like a thermostat for aging. They read the room, nutrient levels, stress, energy, and adjust the cellular machinery accordingly.
Resveratrol whispers encouragement, not.
Gives them fuel, and fasting sounds the alarm. When all three align, they can reset the aging clock just enough to tip the scales toward health, resilience, and perhaps a little more time